Hi, my name is Brendan, and today I'm going to be talking about Clanoflagellates and the origins of multicellularity. This is mostly based on Nicole King's work, her video on iBiology.com, which I link at the end of this. So let me pull this up, if it will cooperate. Boom. Bam. Okay. So I'm talking about animal multicellularity here, basically the evolution of animal multicellularity. I'm talking about essentially organisms becoming multicellular. Life started out single cellular, which I'm going to talk about in a second. And there has to be a path to animal multicellularity. This had to have evolved, right? So Clanoflagellates, these single-celled organisms that are really, really cool, happen to give us a great insight into how organisms started to go from single-celled to multi-celled beings. And so first we got to look at the origins of life. So life originates from one common ancestor. We know this due to conserved sequences across species. All life uses the same genetic formula as well. The adenine, thymine, cytosine, guanine, we all use that same genetic formula for our genes. Every organism on Earth uses the central dogma of biology, which is DNA to RNA to protein. So every cell does transcription, every cell does translation, every cell does protein. This includes plants, this includes fungi, this includes animals, which are all multicellular, as well as all single-celled prokaryotes and eukaryotes. Everything does this. So we know that because everything does this, that we all had to have had a common ancestor, right? And so we know that we evolved from unicellular organisms as well, because we share things in common with all these ancient single-celled organisms. So here we hypothesize that there has to be a transition to multicellularity, single cell to some sort of like aggregate of a bunch of different single-celled organisms. Eventually, these are making all the animals and plants and fungi that we see here. So single cells combine to make one functional organism. If you've ever seen Cells at Work, which is a great show, or probably the greatest piece of cinema in human history, Osmosis Jones, you will see they give a pretty good depiction of cells living in a community. Different cells having different jobs, right? Cell differentiation. These cells start out as stem cells, and then they become, your genes say, hey, do this job. We look at Cells at Work. We see representations of white blood cells, of red blood cells, of the different T cells, killer T cell, regulatory T cell, helper T cell, memory cell, mast cell, all these different cells, right? Osmosis Jones, same thing. Osmosis Jones himself is a white blood cell. He's a cop. He's a cop in the city of Frank in the human body, right? So we get this idea here that single cells are clumping, right? Evolution of clumping here. And we get undifferentiated multicellularity, meaning single cells just coming together and kind of living together and communicating. But then we have a division of labor, differentiated multicellularity, which is what we see in animals, plants, and fungi. Interestingly enough, though, this has evolved multiple times. I actually thought before watching this video, this Nicole King video, that multicellularity would have evolved once, but it didn't. It evolved multiple times, and that's represented, as she talks about here. It's represented kind of where these colors start here. So the metazoa, animals, fungi, plants, right? Multicellularity evolved different times here. And we have a unicellular organism that we kind of share with fungi. Turns out that they're more related to animals than plants are. And then we have one unicellular organism here that evolved its own path to multicellularity, its own genes for cell communication and intercellular interaction, right? Because these cells are living in a community. They're living in a community and they're functioning as one organism. But plants, fungi, and animals have all evolved different ways to do this. This is also represented here, where these genes down here are actually the ones that are involved in basic cellular processes like transcription, translation, and repair, things that we were talking about earlier that all things share in common. that is depicted down here. The genes for multicellularity essentially are going to be actually in here. They're going to be different genes for the slime molds here, fungi, animals, etc. So actually, it turns out that this little group of animals called Clanoflagellates actually would lie right here. So they are pretty related to us. So what are Clanoflagellates? They are these cool little organisms that have they're single-celled microbial eukaryotes defined by a cell body. They have a choano or a collar, right? Choano means collar. So they have the cell body right here, collar right here, and a flagellum. And they're about the size of yeast cells. They feed by using this flagellum. They move by using the flagellum, but the way they feed is they propel this flagellum. They oscillate it a bunch, feeds water into the collar, and this is how they feed on bacteria. They're very diverse. They come in many different forms. Genomes have been sequenced, and they share many genes with animals, genes that previously we did not know that they shared with us. Almost all have single-celled phases to their lives, but some can form aggregate colonial structures, meaning they all come together and kind of communicate and operate as one entity, such as this Rosetta species right here, which is depicted here. They form these really cool spherical rosette shapes, right? So these guys are super, super important. They form these colonies using this Rosetta kind of inducing factor, they form these colonies in the presence of this Rosetta inducing factor, is what I meant to say. So what is that? It's RAF1 here. Sulfonolipid produced by prey bacteria, Algoriphagus machiponganensis, I think is how you say that. That triggers colony formation in this Rosetta species. This indicates that certain prey, so these are prey bacteria, triggered colonial behavior in these organisms. They're connected by these intercellular bridges here, as you see here. So they physically connect. They communicate, and they form an extracellular matrix, this kind of water kind of matrix that they form when they're all together in between all the cells, and they use this for communication. This is what animals do as well. Plants and fungi, they have extracellular matrices for which these communications happen. And we see this depicted here. We have a slow swimmer rosette species. Presence of this bacteria, the RAF factor here, that is produced, induces formation of rosette colonies. They are forming like an animal-like kind of entity here. And this is why this is super important, because this resembles choanocytes and sponges actually. So sponges, how they work essentially is they move water through their central cavities and they filter feed. You can watch videos on YouTube, which I highly recommend, of sponges actually like divers putting dye near sponges, and the water moves through the sponge, and you can actually watch as these sponges actively move water through. And these choanocytes here, so this is how the sponge eats. You can already see the similarities. They look a lot like our friends, the Clanoflagellates, right? So, and they're operating much the same way. So they're called collar cells. Remember, choano, collar. So remember, this flagellum is basically shifting water into these collars, and this is how they're feeding. And here sponges exhibit bactivery, so they are eating bacteria, much the same way that Clanoflagellates do. And here we have these cells communicating. They're all communicating, much like the Rosetta species. And we see a cell undergoing apoptosis here. So they're communicating. They're going through programmed cell death. Maybe another cell was like, hey, you need to die. Like, you got a mutation. And the cell's like, okay, cool, I'm gonna die, right? And then this happens. And here we have somatic cell differentiation. So here we're seeing an animal here. We're seeing in animal form. You can see in this expanded view, the choanocyte looking a lot like a Clanoflagellate, much the same body plan here. So, and this is how the sponge feeds. And this is how we believe multicellularity has evolved. This is what Nicole King's findings suggest here. And she does talk about sponges in her video. So the key takeaways here, so I think this is a really cool subject, but key takeaways here, genetics allows us to get a glimpse into animal origins of multicellularity by comparing the genomes, right, and finding these conserved sequences. That is how we gained so much of this understanding here. And this is how Nicole King herself actually was able to make these studies, right? Understanding these origins gives us better insights into how our own bodies function at a cellular level, how we evolved, how sponges evolved, how animals evolve, and all these advanced because of this. And the cool thing here is multicellularity evolved multiple times. Convergent evolution, right? These the idea that these organisms are evolving the same thing, but at different times under different selective pressures. Super, super cool. So anyways, that's my video references here. I thought it was really cool. This was a really cool video. Check out that video. It was sick. And yeah, thanks for watching. That is it.